what we've also done is systematically record from several neurons, excite one neuron, which is the action potentials evoked here, and record. Everybody knows it's an electrochemical transduction process. You produce a little analog response in the target cell. And what you will notice here is that from this cell onto this cell, you have this rapidly shut down in the transmission. In a simple sense, this can be seen as a low-pass-like filter, but it's really a nonlinear dynamic synapse. You can also have synaptic transmission onto a different kind of cell where you have rapidly facilitating synapses, and it can be seen as a high-pass-like synapse. Now, it turns out, so every neuron contacting other type of cell has a unique nonlinear synaptic property. So we had to derive for all the major pathways the exact types of dynamics that we're going to use to model the transmission accurately between the different types of cells. So there is a map, and there are specific rules, and we've not been able to record from every one of the several thousand pathways, but we've derived rules that allows us to generalize to those that that have not been recorded. You also have maps for the inhibitory inputs, and these allow us now to systematically model the different connections between the different neurons in a biologically accurate manner. This is just more or less what a mapping looks like. You have different classes of synapses, and they're assigned between different types of cells. There's a lot of other parameters, biophysical parameters and physical parameters, that are needed to capture the model so that a neuron, when it fires, will inject the right amount of current at the right time into the target cell, and those have been derived. And we can now go to the circuit builder. Once we've connected it and we've assigned who is going to have synapses with it, we can now assign all the synaptic properties which get inserted into an algorithm that will act as a synapse. And this just shows that we can actually, through the model, capture all the different types of dynamics impinging on the cell. All of these cases, about 30 hertz stimulation of the source cell, and you can see that you have the whole spectrum of different types of dynamics that is synaptic injection into this type of cell. So we can now capture the detailed synaptic dynamics between all the major types of cells in the neocortex. There are many other things. We've also looked at learning algorithms, and this is the spike timing dependent algorithm, which we showed many years ago. In addition, there are many other algorithms, because it turns out that you have to, if you have 20 million nonlinear synapses, you need learning algorithms that are going to align them and adjust the filter functions. And we've done a lot of experiments deriving some of these learning algorithms, and they will be implemented. So at this point in time, we do believe that we have the starting building blocks to a sufficient level of biological detail to begin. It's just to begin. It's to take the first step in building the circuit, simulating it, and eventually visualizing it. So, of course, what we do is we take each neuron. You need a lot of neurons. We are simulating for the first time thousands of neurons, that each neuron has the full morphological and electrical complexity. And so the simplest way is just to put one neuron in each processor, turn a processor into a neuron, and use the MPI messaging as axons. And that's essentially what we do, and we've written 2 software packages that allow us to do this kind of simulations. But the problem is that it produces about 1 terabyte of data in 1 second of simulation. So we needed another supercomputer, because we want to decide which terabyte we should keep for analysis. We don't want to spend 1 second of simulation and then spend 3 weeks or a year or half a year to analyze it, and then decide this is nonsense and start another simulation. So we want to use a visualization to immediately allow us to assess whether this is something that is interesting. We want to store it, we want to save it, analyze it further. And so we went to SGI because we needed very large amount of shared memory. And they were very nice in that they gave us a 90% discount on this little supercomputer. And this allowed us now to build a small media center in Lausanne, where we can create 3D representations with glasses, and we can sit inside this neocortical column. So it's more like, like feeling like a child. You want to build this puzzle and you want to sit inside and actually see how the circuit works. I'm not sure if it's for science or for pleasure, but it is a lot of fun. So this is a first simulation. Now bear in mind, this is very crude. It's a first simulation of 10,000 neurons, and we're only visualizing 10% of them. And the different colors are the different neurons as they fire. And the visualization was really done in the last 2 weeks. So this is really the very first version of the visualization, and it's going to get 1,000 times better than this. But what this just gives you a feeling of the immense complexity of what you could, in principle, see in terms of having the different parts firing. If you watch carefully now, you'll even see action potential propagation. You see that cell propagates. Another one is going to, the voltages propagate. So you really can now zoom in onto single cells and watch the electrical activity emerging. We can screen and we can select and say we only want to see a certain type of cell, or we want to see a certain ion channel, or we want to wipe out a certain type of neuron and see what's the effect on the circuit. There are 1,001 in silico imaging experiments that can now be done in a very rapid, with very rapid time scale, and that can generate hypotheses for studying the circuit further. So I'll wait for this just to give you a quick view of what it looks like to dive inside. Now we're looking at the top of layer 1, and we're going to go inside while the circuit is active. And if you can see at the top some of the voltages changing in these branches. And this, of course, is one thing to realize is that this circuit is still not calibrated. This is what we call version 2. We're aiming at, by the end of the year, only reaching version 10. So this is not calibrated, but it is the first functional 10,000 neurons with activity, electrical activity, and so on. This is just a single cell. You can extract this one cell out of the 10,000 and decide you want to study just what is happening to one cell. And here you can now zoom in and start slowing down. And if you see interesting things happening, and you could relate that to, for example, synapses changing and so on. Okay. So this is just an example of what we call a biological. Everybody says it's impossible. Why do you do it? There's so many things you don't know. Well, our philosophy is, if you don't know it, don't let it stop you. Okay? So you begin. If you don't know it, you assume it, and you find ways to do experiments to fill in those gaps. So we have the first version, and we're already on version 2, which is a different level of refinement of the biological data. And we're going through progressive steps until we will reach version 10, which will capture as accurately as we can the data that we've collected over the past 10 years, and that should capture a tremendous amount of data that is known in the literature for the past 100 years. There are other things that are needed to be done. You can build the circuit, but how do you calibrate it? So we can calibrate and check each of the parameters, but we also need to calibrate its function. We don't want to put this into a behaving brain, because you can then calibrate anything you want. We want to calibrate that the circuit itself functions correctly. And what we start doing is we do experiments of where we bounce information between neurons. So we record from 3 cells, we stimulate it, it activates another cell and produces another cell. And these disynaptic pathways allow us to start working out properties that if we do the same thing in the simulation, we should see similar statistics of firing other cells, how they fire them, the amount of current that is injected into another cell via another cell. But we can do that now with a 12-patch to an even more sophisticated level, allowing us to look at the circuit and to look at circuit statistics to see that we get the same. These are these little disynaptic pathways that are activated through a second cell. These are direct connections. But we can also do a whole host of dose-response curves where we activate a set of neurons and look at one neuron and see how it behaves.